Okay, so in this video today, we're talking about fermentation. So this is a process that happens in cells when oxygen is not present. So this is an anaerobic process. And this process allows organisms to repeat glycolysis, so to keep completing glycolysis, even when we can't continue the rest of aerobic cellular respiration. So remember, glycolysis still results in 2 ATP per glucose. So the benefit to fermentation is that we're able to still make at least a small amount of energy even when oxygen is not available. And so there's two types of fermentation. There's actually more than two types of fermentation, but we're just going to focus on two that are kind of more common and relevant to us, which are lactic acid fermentation and alcoholic fermentation. So after glycolysis, so glucose comes into the cell, we go through glycolysis, we create two molecules of pyruvate. And pyruvate is kind of a branching point. So if we have oxygen, or if we don't have oxygen, we're going to do fermentation. We're going to make ethanol, so alcoholic fermentation, or we're going to make lactate through lactic acid fermentation. And again, that's anaerobic respiration, so in the absence of oxygen. If there is oxygen, then that pyruvate is going to go to the mitochondria, and it's going to enter the Krebs cycle, and then the carbon dioxide is going to get released, and the electron carriers are going to go and participate in the electron transport chain. So that's aerobic respiration if we have oxygen. So this is especially important in our muscle cells. Our muscles need a lot of energy. So if you've heard of like aerobic and anaerobic exercise, our muscle cells are often one of the first to run out of ATP and therefore to have to do fermentation more often. So you hopefully remember that NAD plus is a reactant in glycolysis. So NAD plus is one of the molecules that oxidizes the carbons in glucose, and that process is what generates some of the electron carriers that then later get used, usually if we have oxygen, in the electron transport chain. So in order to continue doing glycolysis, we have to have all of these reactants, which means we have to have a source of NAD plus. So if we don't have oxygen and we do glycolysis, if we run out of NAD plus, we're no longer going to be able to do glycolysis anymore. So generally speaking, in order to recycle NADH to NAD plus, it has to drop off its hydrogens and electrons somewhere. So here's our pyruvate molecule. When we do have oxygen, this is what we've been talking about kind of over the last week and a half. If we do have oxygen, then that NAD plus drops off electrons or the NAD plus picks up electrons in the Krebs cycle, and then in the electron transport chain, it drops it off again, so we get NAD plus and we can repeat the cycle. If we don't have oxygen, then we have to do fermentation, and so we're either, depending on what organism you're either going to take that pyruvate and form lactate, and in that process we're actually adding hydrogens back to the pyruvate molecule, so we oxidize the NADH and we remake NAD plus. Alcoholic fermentation is similar, so we actually end up adding the hydrogen back to the pyruvate product to make ethanol, and that again oxidizes the NADH back to NAD plus, and so we can then use that to continue glycolysis in the absence of oxygen. So that's lactic acid fermentation. This is alcohol fermentation. So the path that you use, so again, generally we do this one with oxygen, but we also have enzymes to do lactic acid fermentation. Just depends on what enzymes you have available to you. So it's very organism dependent. A lot of bacteria and yeast are going to do alcoholic fermentation. So they recycle their NADH back to NAD plus by taking that pyruvate and converting it to ethanol. And then that NAD plus can go back to glycolysis. We utilize this process to make beer and wine and bread. So bread. Lactic acid fermentation, on the other hand, this is what we do in our cells and also what some fungi do in their cells when they don't have enough oxygen. In that case, we recycle our electron carrier by converting that pyruvate into lactic acid. So, and then that can then go back and keep doing glycolysis. And so this is also what we use to cheese and yogurt and our mussels there. So we're going to spend a little bit of time on each type. So you should be able to kind of compare and contrast the two processes. Alcoholic fermentation, again, we're taking pyruvate, we're adding a hydrogen back to it from their NADH, and that's going to make ethanol and carbon dioxide. So you can see here in the diagram, we go through glycolysis, we make pyruvate, and that pyruvate goes through a series of reactions where actually one of the carbons breaks off. So we do also make carbon dioxide in this process. And then we actually add a hydrogen back to the carbon here. Or sorry, we make this, we don't add the hydrogen back yet. We get this compound, and then the NADH actually adds a hydrogen back to actually the oxidant. So that's what makes that hydroxide group, makes ethanol and alcohol. So couple important things to know about alcoholic fermentation. This is a dead-end process. So once we make this ethanol, we aren't able to convert it back to another usable form in our cells. And it also can be fatal to organisms to have too much alcohol around. And so yeast can do this for a certain amount of time, but then if they make too much alcohol, then they're going to poison themselves and die. Additionally, you can't, and like I said, you can't reverse this reaction, so you can't go backwards. But always remember, we have our conservation of mass here. So our pyruvate atom has 3 carbons, ethanol only has 2. That third carbon got released as carbon dioxide. So in yeast, so you can see this process in yeast because the carbon dioxide is actually what makes the bubbles in the bread and makes the carbonation in some alcoholic beverages, although bacteria is also involved in that process. Lactic acid fermentation, again, the point of this is to oxidize our NADH and convert it back to NAD+, so we can continue to do glycolysis. And in this diagram here, you can see we do glycolysis to make pyruvate. And then you can see here where the hydrogen is being added back on. And so then we now have NAD+, and that can go back, get it back, and we can continue to complete glycolysis. So this, unlike alcoholic fermentation, this is a reversible process. So this lactic acid is eventually going to make its way to our liver. Actually, within a couple hours of like heavy exercise, that lactic acid is going to be out of your muscles into your liver. And it can get converted back to pyruvate. And then when we do have oxygen available, that pyruvate can then continue on to complete the rest of our aerobic respiration process. So once oxygen's available, that lactate's going to be converted back to pyruvate in the liver. So again, in this case, pyruvate has 3 carbons, lactic acid has 3 carbons. When we do have oxygen, we can convert that lactic acid back to pyruvate. And then we can continue with that pyruvate can go to the mitochondria and can participate in the Krebs cycle, etc. So for the third time, pyruvate molecules are converted to lactic acid to get NAD+ back for more glycolysis. This is the process that we use to make like a sour milk, cheese, yogurt, sauerkraut. Some of those processes, so there are also bacteria that do lactic acid fermentation. So some bacteria do alcoholics, some do lactic acid, some do other forms of fermentation that we're not going to talk about today. So if muscle cells are really active, and so this is where the term anaerobic exercise comes from, they can make lactate temporarily. So this is actually questionable, but you can kind of think, your muscles burn when you do a lot of excessive work because you're forming that lactic acid. Again, that lactic acid gets cleared out of your muscles after a couple hours. So when your muscles hurt, if you've done a lot of exercise you're not used to doing, etc., that's probably more likely that you're tearing muscle fibers, and so that those will just take some time to reheal. So that's the lecture on fermentation. Let me know if you have any questions. Thanks for listening.